Hello, good afternoon. Uh, this is the sixth time that we have the unsupervised machine learning seminar, and I want to welcome you all. It is done at Afeka College of Engineering, and we're organizing at ACLP, Afeka Center of Language Processing. We think that unsupervised machine learning is a crucial part of machine learning these days, and it will be more and more important. So thank you again for all who join us on this webinar, and I hope that next year we will be all together, not by web, but sit and chat and talk together. Our keynote speaker and our guest is Professor Brendan Murphy from University College of Dublin. For a long time, we tried to have Professor Murphy with us, and we are very happy for this opportunity. Brendan is the head of the Department of Mathematics and Statistics. He is currently editor for Social Science and Government for Annals of Applied Statistics. Brendan's research interests are clustering, classification, and latent variable modeling. And application interests are social science, food science, and medical and biology. His keynote will be model-based clustering with sparse covariance matrix. Welcome all, Professor Murphy. And just one note, you can ask questions or by raising hand or by Q&A, and I will read the questions. You can choose. I will try to give opportunity to most of you to ask when the time will come at the end of the keynote.
Okay, so today I'm going to talk about model-based clustering with sparse covariances. This is work that I have done with Michael Fopp and Luca Scruca, two Italian collaborators. Michael Fopp is now a colleague of mine, and Luca has been a long-term collaborator of mine in the area of model-based clustering. I'm going to start by introducing model-based clustering, just an outline of the talk. So I'll introduce model-based clustering, and I'll discuss issues with model-based clustering when you have high-dimensional data. I'm then going to talk a bit about graphical models, and in particular graphical models related to sparse covariance matrices. And then I'm going to talk about a model that we propose for model-based clustering called mixtures of Gaussian covariance graphical models. And then we're going to look at some applications on real data, in particular on some data to do with breast cancer, molecular subtypes, and then I'll have some discussion at the end, and I'll talk about some results and software that we have for the methods that I presented in today's talk. So model-based clustering, in its most basic form, is an approach to doing cluster analysis, which cluster analysis is when we're looking for subgroups within a data set or within a population, and in particular, when we are looking for capital K subgroups. And I'll discuss about the choice of K later. In model-based clustering, we assume that the data comes from a mixture distribution, and most commonly, people assume a mixture of Gaussians. And we also make the assumption that each component in a Gaussian mixture, each component of the mixture corresponds to a cluster. So to make this more concrete, suppose we have data points xi, where each is a v-dimensional vector of values. So it's a v-dimensional realization of some random variable. And the model we're proposing for xi, so the distribution of xi, given the model parameters, is made up of a sum over capital K components. Tau k is the probability that an observation comes from component K. And then we model data within component K, using a multivariate Gaussian distribution with mean UK and covariance sigma K. So then when we integrate, because we don't know the cluster membership of each observation, when we sum over the probability of an observation coming from each group and the distribution of the data within each group, we get this mixed form of the distribution, which is a mixture of Gaussian densities.
Here's some information.
Oh, sorry, clicked the button. Just want to get rid of a window. Sorry about that. I clicked something I shouldn't have. So once we've fitted a mixture model to our data, if we have an observation, we can estimate the probability that the observation came from cluster K, given the observed value, using Bayes' theorem, and it comes out to be tau K times the component density divided by the mixture distribution. So that's a useful thing about when you do model-based clustering, you get a probabilistic assignment of each observation to each cluster. You could do model-based clustering using a number of approaches, but what we find to be most efficient is to do model estimation by maximum likelihood or penalized maximum likelihood using an EM algorithm. But you could just as easily do a Bayesian analysis to do model-based clustering, but there's a lot of extra kind of overhead in doing that. And sometimes that overhead doesn't really pay off in terms of results. Other times it does, but we find a very pragmatic way of doing model-based clustering is to use the EM algorithm for model fitting. One of the problems when we're doing model-based clustering, I'm after losing my window here, trying to get rid of something that's sitting on top of my screen. Just need to do something here, sorry. There's a, one of the problems we have with model-based clustering when we fit a mixture of Gaussian distributions is that the covariance matrix that we have in our model is quadratic in the number of variables. So as the number of variables grows in your data set, the number of parameters in your model explodes very, very quickly. And here's just an example of the number of parameters in a Gaussian mixture model as a function of the number of clusters. And you can see that it grows quadratically, and then the steepness of the quadratic will depend on the number of components in the mixture model. So very quickly, we end up having a dramatic over-parameterization in the model. We've too many parameters to estimate from our data. Even if we can fit a model, it tends to be numerically unstable. And we also, when we fit a model to high dimensional data, we find that the models tend to overfit the data, and that can cause problems with the classification results that come out of the clustering. So when you estimate those cluster probabilities, they tend to be very close to 0 or very close to 1. So we don't really capture the uncertainty in any clustering. A number of solutions have been proposed for this, and I've actually worked on a number of these solutions with different collaborators, and lots of other people have come up with other approaches. One approach for doing this is to consider the covariance matrix and look at its structure very, very carefully, in particular its mathematical structure, and consider different covariance decompositions to see, are there methods for reducing the number of parameters in the model? Another approach is to basically, even though we have v-dimensional data, maybe the v-dimensional data that we're observing is really just some noisy manifestation of some lower dimensional kind of random process. So that's what I mean by dimension reduction and factor models. And I'll be talking about these in turn later. And then another approach that's been proposed is to take our data set and to do some variable selection to reduce its dimensionality, either before we do clustering or as part of the clustering process. So in machine learning, if you do it before the clustering, a lot of people will do that. They may do something like principal components analysis on the data and then do clustering. But there are problems with that approach because sometimes when you do your your variable selection or your data reduction before you do your clustering, you may actually lose important clustering information. So sometimes it's a lot better to do it as part of the clustering process. So the first approach I'm going to talk about that people have proposed to try and allow for model-based clustering using Gaussian mixture models for high-dimensional data is based around the idea of the eigenvalue decomposition of the covariance matrix. And there's a software package called mClust that's written for OR, the OR statistical software package that implements this in a very comprehensive way. So we're going to do a slightly different version of the eigenvalue decomposition of the covariance matrix here. So we're going to write the covariance matrix in terms of some scalar, lambda. D is a matrix, an orthogonal matrix. A is a diagonal matrix, where the elements of A are proportional to the eigenvalues. And then we have D transpose, which is the transpose of the orthogonal matrix we had earlier. We restrict A to have determinant 1. So basically, it's different to the usual eigenvalue decomposition in terms of we've taken a constant out of the usual eigenvalue decomposition and taken that out of the front of the expression, and then the values of A are proportional to the eigenvalues rather than being the eigenvalues themselves. And this matrix is constrained to have determinant 1. This decomposition, even though it just seems like a mathematical curiosity at the moment, has a very nice interpretation. The value of lambda controls the volume. So a Gaussian distribution, the contours of a Gaussian distribution are elliptical. Lambda controls the volume that the Gaussian distribution takes, basically. So how scattered is the data? So if lambda is large, we get a cluster that's very diverse and scattered. If lambda is very small, we get a very tight cluster. So for example, the green cluster I'm highlighting here with my mouse, I don't know if you can see that in the VEI model, is very large. So that would have a large lambda, and the blue one would have a low value of lambda. The D matrix, the orthogonal matrix, controls the orientation. And A controls the shape of the ellipse. So if the values of A are almost all equal to each other, we will get, or if they're exactly equal to each other, we'll get a circular cluster. If they're close to being equal to each other, we will get something that's very close to being circular, but a quite circular ellipse. And if the values of A are quite different from each other, we're going to get an ellipse that's elongated in some directions and very squished in some other directions. So the three elements of this eigenvalue decomposition of the covariance matrix have very nice interpretations. Lambda controls the volume, A controls the shape, and D controls the orientation. We then have the option of assuming, to reduce the number of parameters in our model, we can assume that some of these terms of the eigenvalue decomposition are either equal or unequal across clusters. So we say we use E for equal and V for variable. So for example, just to take an example, if you look at the 14 panel plot below, the VEV model assumes that we have clusters that have different volumes. The first letter is volume. The second letter is shape. So all three clusters have the same shape. And then the last letter is V. So that means that they have variable orientations. So they're allowed to have different orientations. So VEV says different volume, equal shape, different orientation. VEE, just to look at that one as another example, we have clusters that have different volumes, but their shape and orientations are equal. So this, by constraining things to be equal or unequal across groups, this gives us a way of reducing the number of parameters quite considerably. It's also worth noticing that some of these panels, in some of them, we have the letter I. The identity matrix is an orthogonal matrix. It's also a diagonal matrix, and it has determinant 1. So we have the option to allow D and A equal to the identity matrix. If you make D equal to the identity matrix, if you look at any of the models where the last letter is I, the orientation is the identity matrix, you'll notice that the ellipses point in the direction of the axes. And if you let A equal to the identity matrix, the shape, you get circular clusters. So we get 14 different models, whether we choose to let the volume, shape, and orientation equal to each other, variable, or equal to the identity matrix. If the last letter is equal to I, so the orientation is equal to I, the resulting covariance matrix is a diagonal matrix. So instead of it having a quadratic number of parameters in terms of the dimensionality of the data, it's either going to be a constant or linear in the dimensionality of the data. So this is one way of dramatically reducing the number of parameters in your covariance matrix. And then you can actually apply Gaussian mixture models to high dimensional data sets. Just to make a small detour to probably a clustering method that's very, very well known, we can make a connection between model-based clustering when we assume the EII covariance structure and k-means clustering. So just to go back to the previous slide for a second, because I think it's important to show you the picture. If we take the M-clus models and we assume that every cluster, the first letter is volume, we're assuming that every cluster has the same volume, the shape is the identity matrix, and the orientation is the identity matrix. We get equal shape, equal volume, circular clusters. If you make that assumption, and you make one further assumption, you actually also assume that the probability of each component is equal. Then when you try to fit this model using a slight variant of the EM algorithm called the classification EM algorithm, you get exactly k-means clustering. The algorithm for fitting this mixture model is using a classification EM algorithm is precisely the k-means clustering algorithm. And that's a very useful insight to know, because it'll explain to you when k-means clustering works very well and possibly when k-means clustering would not work very well. So k-means clustering actually is fitting a Gaussian mixture model where all of the mixture components get the same weight and where all of the mixture components have identical covariance matrices that are just a constant times the identity. Another approach that's used for high dimensional data is to assume that the covariance matrix sigma k can be decomposed into the following form. We have a V by Q matrix called lambda k, and they're called the loadings, that we can write it as lambda k transpose plus some diagonal matrix psi k. This covariance structure arises very naturally in factor analysis. And basically, implicitly, what you're assuming when you have this covariance structure is that the data within each cluster lies on a very close to a Q-dimensional plane in V-dimensional space. How close to that plane are we lying in the higher dimensional space? So effectively, the data has an intrinsic dimension that's a lot lower than V. Q is kind of the dimensionality of the data within each cluster. And then the psi matrix is telling us how close to that plane the data actually lies. So that's kind of a interpretation of where the factor analysis structure comes. A useful thing about this model is the covariance matrix for a fixed value of Q ends up being linear. The number of parameters ends up being linear in the dimensionality of the data. So we actually end up getting a covariance matrix that is not necessarily diagonal. So it doesn't necessarily have the clusters pointing aligned with the axes. And it's still, the number of parameters are linear in the dimensionality of the data. So basically that's a nice feature of the mixture factor analyzers model. this covariance structure has been exploited very, very often in the mixture of factor analyzers model, which was Gahramani and Hinton proposed that model in 1997. There were models by Christopher Bishop and Mike Tipping in Microsoft Research. They used a version of this model called the mixture of probabilistic principal component analyzers, which is actually this model where the last term, psi k, is just a constant times the identity. And they showed that when you fit this model, you're fitting something very, very close to factor analysis within each cluster. And I did work with Paul McNicholas, one of my PhD students, a few years ago, where we developed a family of models called the parsimonious Gaussian mixture models, where, like the M-CLUS models, we decided to allow some of the elements of this covariance decomposition to be equal or unequal across groups. So we allowed for lambda k to be equal across groups or not, psi k to be equal across groups or not, and then we allowed the assumption that psi k was a constant times the identity. So that's like an isotropic constraint. So we ended up coming up with a family of models by considering all possible versions of those constraints. And that was another way to reduce the dimensionality of the model space, reduce the number of parameters, and thus improve clustering and classification performance and make model estimation much more stable. So these are two approaches that we've considered initially to try and reduce the dimensionality of the data. In the work I'm about to start describing, we're going to take quite a different approach to this. So the two approaches I've talked about so far, they don't kind of automatically directly get you a sparse covariance matrix. So in a mixture factor analyzers model, yes, you've reduced the number of parameters used to describe the covariance, but when you estimate the full covariance matrix, it could be that every element in the matrix is non-zero. Some of these models are a little bit difficult to interpret when you're using them. They may give good clustering results, but the final covariance matrix doesn't have a clear interpretation. And you don't really have a direct way of modeling the relationships between variables within clusters. In the mixture factor analyzers model, you can see it from the connection between you can see it from the loading matrix, the relationship between the variables, but it's quite an indirect way of seeing the relationship between variables. I should also emphasize in the M-CLUS models, the only way we can really get the covariance matrix to be sparse is if we assume that the D matrix, the orientation matrix, is the identity. And if we do that, we end up getting a diagonal covariance matrix. So the M-CLUS family goes from having a full covariance matrix to having a diagonal matrix in one step. So that's quite a big jump, and there's nothing really in between the two. You can go from having a diagonal matrix, which is linear in the data dimensionality, to having a full covariance matrix, which is quadratic in the data dimensionality. So just to even show a very low dimensional small example, assimilated data, just to show what we may want to achieve. Here's a two-dimensional data set. And what I want to do is, we've two clusters. In cluster one, we just have a cluster of points. The ellipse is pointing in a kind of up towards the right-hand corner of the plot. So the two variables are clearly correlated. And we have a full covariance matrix, which is sigma 1, 1, given that we belong to cluster 1, sigma 1, 2, sigma 1, 2, and sigma 2, 2. So to model the circular points in the top right-hand corner of the plot, we need a full covariance matrix. But to model the points in the bottom left-hand corner, the two variables are uncorrelated, so having a diagonal covariance matrix would be appropriate. So what we want to do is we want to come up with a model-based clustering approach that's going to reduce the number of parameters in the covariance matrix for each cluster and set them equal to 0, so that basically we reduce the number of parameters in our overall model in a dramatic way. and that will improve the stability of our inference and hopefully our clustering results as well. The way we're going to do this is using something called a Gaussian graphical covariance model. So a graph is a mathematical object that has a pair. So the graph G has a pair. It has a set of vertices and a set of edges. For a Gaussian covariance graph model, what we do is we are going to assume that we have our data follows a multivariate Gaussian distribution with some covariance, and the non-zero elements of the covariance are described by the graph. So looking at the picture I have here, I have 5 variables, x1, x2, x3, x4, and x5. And I have an edge between x1 and x2. I have an edge between x1 and x3. I have an edge between x1 and x4, an edge between x2 and x4, and an edge between x3 and x5. So they're going to be the non-zero elements of my covariance matrix. So the covariance matrix has a diagonal, which has to be positive. And then I'm saying that my covariance matrix has non-zero covariance between x1 and x2, x1 and x3, x1 and x4, x2 and x4, and x3 and x5. And all the other elements that are blank in this covariance matrix are going to be constrained to be exactly equal to 0. So we're after directly assuming, given a graph, we have a direct sparse structure in our covariance matrix. So all the missing edges in this graph, so there's no edge between variable 3 and 4, so we're going to get a 0 in the covariance matrix for the covariance between variable 3 and variable 4. So if you have a multivariate Gaussian distribution with this structure, that's called a Gaussian covariance graph model. This differs from a lot of people when they're looking at Gaussian models with sparsity. Instead of directly modeling the covariance matrix, they will model the inverse of the covariance matrix, which is called the precision matrix. And they will assume that the precision matrix is sparse, where we're actually taking the approach of directly assuming that the covariance matrix is sparse. Now, we're interested in clustering in a lot of the work that we do. So what we're going to do is we're going to assume that we have a mixture of k Gaussian densities, but then we're going to assume that each Gaussian density is a Gaussian covariance graph model. So it's different from the previous Gaussian mixture model because we have this graph GK, which is telling us which elements of the covariance matrix are allowed to be 0 and non-zero. And the covariance matrix is thus constrained to be in the cone of positive definite matrices that have zeros and non-zeros in the correct locations, which are described by the graph GK. So this is a new family of Gaussian mixture models for clustering that is going to allow us to get sparse covariance matrices if our graph has very few edges, and thus we may be able to model high dimensional data with bigger accuracy, basically. So that's the formulation of the model that we're going to be looking at. We're going to we can't just fit the model directly. We want to do two things. We want to estimate what graph would be appropriate for our model. And then given the graph, we want to fit a Gaussian covariance graph model. So what we end up doing is we end up setting up a penalized log likelihood for our model. So the first term in the log likelihood is the log likelihood for a Gaussian covariance graph model, a mixture of Gaussian covariance graph models, if we knew the graph that was being used to tell us where the 0 and non-zero elements are in the covariance matrix of each group. We're also going to want to estimate the graph. So which graph would be appropriate to get a good model for each group in our data. So what we're going to do is we're going to put a penalty on our likelihood, and we're going to penalize graphs that are overly complicated. So we're putting a penalty on our likelihood where we put, I'm going to go through some explicit penalties in a second, where we have a function Q that takes in the adjacency matrix of the graph. So it's a binary matrix that tells us which pairs have edges between them and which pairs don't. And we're going to put a penalty on the adjacency matrices. So if the adjacency matrix has too many ones, the penalty is going to be large. And we're going to have a separate penalty for each component of the mixture model. And then we're going to build a version of the EM algorithm, which is going to allow us to estimate the appropriate graph for our data and then estimate the models of the Gaussian covariance graph model for each component, assuming the graph and then the mixing components as well. So we're going to estimate all of the parameters and the graph in one EM-like algorithm for fitting this model. And such an algorithm is called a structural EM algorithm. So once we know what graph we're using, the algorithm reduces to a standard EM algorithm, but then we have an extra step in our algorithm to estimate the structure of the graph that's been used to describe the covariance matrix of each component in our mixture distribution. So how would the algorithm work? If we assume that we know the graph, the E step of the EM algorithm is exactly the same as a standard EM algorithm. We have our current estimate of the graph for each of the mixture components. We have an estimate of the covariance matrix for each of the mixture components that has non-zero elements in the positions that the graph has edges. And then we have the mean of each mixture component, and we have the probability that an observation comes from each mixture component. So what we're doing in the E step is we're estimating what is the probability that observation I comes from component K. So this will be a probability between 0 and 1, and when we sum across all capital K components, we get that these terms sum to 1. In the M step, we want to maximize the complete data log likelihood, which takes this form. So now we have the estimated probability that an observation comes from each component in our likelihood. And then we want to maximize this expression. And we're going to maximize it with respect to the parameters tau, mu, and sigma, and with respect to the graph, G, which has adjacency matrix AK. Estimation of tau k and mu k is straightforward. Tau k ends up being as just basically working out what proportion of observations belong to component k. The maximum likelihood, the estimate of mu k on each iteration is just what's a weighted average of the observations that belong to, where the weights are given by the probability that an observation belongs to component k. We then need to search for a graph and then fit the covariance matrix that has non-zero elements where the graph says they should be. For the S step, we need to search over all graphs, basically. What we do is we've actually done two approaches. I'm going to describe one approach. We used a genetic algorithm to search over the space of all possible graphs that describe where we have non-zero covariance terms, where we want to have them in our model for component K. So we have a population of graph structures. So we have capital O graphs that we're considering to use. For each of those graphs, we estimate a covariance matrix that has non-zero elements in the correct place for that graph. And we do this using iterative conditional fitting. There's a very nice algorithm that was proposed by Sanjay Chowdhury and Matthias Dirton. I have a reference for it at the end of the talk, where it allows you to estimate a covariance matrix with zeros in prescribed locations. And we can do that to estimate the covariance matrix for each group. We then evaluate our objective function, which I had on the last slide, and we choose the fittest solution. So we choose which of the population is best. Then we do some genetic operators on that to form a new population, and then we keep iterating this algorithm. So it's a genetic algorithm for estimating the graph that is best used for modeling the data within each component. When we submitted this paper for publication, the referees suggested that when we had a lot of variables, the genetic algorithm would become quite inefficient. So when we were revising the paper for publication, we also developed a greedy algorithm for searching over graphs. So, and that scales to much higher dimensions than the genetic algorithm, which may be more accurate. So I'm only kind of mentioning the genetic algorithm here, but there is also a greedy algorithm that scales for higher dimensional problems that we developed at the same. Well, a nice output from the refereeing process of an article that we actually kind of made our method more useful in the process. We also included prior distributions to avoid singularities, basically, and kind of instabilities in our estimation of sigma. So we used prior distributions to regularize the problem. So we actually, rather than doing maximum likelihood, we actually did maximum a posteriori estimation. So we put a Dirichlet prior distribution on the mixing proportions. We put a normal prior distribution on the means of the mixture components, and we put an inverse Wishart distribution on the covariance matrices. And then we also put a prior distribution on the graphs that depended on the penalty term, basically. And that just avoided some kind of singularities that you can get when you're doing fitting mixture models in high dimensional spaces, where a small number of observations ends up in one cluster, and then the covariance matrix becomes ill-conditioned. Now, up to now, I've ignored the penalty functions that we use in our estimation. So the first thing we thought about doing, and a lot of the penalty functions are going to depend on the number of edges in the graph. So if we take one of our graphs that we're using to describe where do we have zero and non-zero terms in our covariance matrix, we can just count up, well, the graph, we can describe the graph as an adjacency matrix. It's a binary matrix where there's a 1 if there's an edge between two nodes, and it's 0 otherwise. And we count up how many edges there are. And because the matrix is symmetric, we only need to count up each edge once. So EK is the number of edges in the graph used to describe component K. If we were to use a Bayesian information type penalty on the graphs, we would just take a penalty that's half the number of edges times the log of the number of observations. And that can be quite effective. It'll discourage us from having a graph with too many edges, because you can see as the number of edges grows, the penalty grows. So when we're doing our structural EM algorithm, this will discourage us from having a graph with too many edges. Another penalty for a graph estimation that's been proposed in the literature is an extended BIC algorithm that penalizes the number of edges in terms of the number of observations, but it also has a second penalty in terms of the number of variables. And you can choose the value of gamma to balance the penalty between the number of observations and the number of variables. Clearly, if you let gamma equal to 0, you're going to get BIC back. And if you let gamma equal to 1, you're putting quite a large penalty on the number of variables. So this will, if you let gamma equal to 1, it's going to get you much more sparse graphs when you're in high dimensional settings, because the number of edges is being multiplied by the number of variables times 2 gamma. So for high dimensional problems, if you want a sparse covariance matrix, making gamma large is going to encourage such sparsity. We also thought about models that people have actually used for random graphs. So when people study random graphs, one of the most famous models is the Erdős-Renyi model. So we were looking at, you know, if we take e to the power of minus a penalty, that's going to give us something that's very like a probability distribution. It's like a Boltzmann distribution in statistical physics. So we were thinking that if we let e to the power of minus our penalty equal to an Erdős-Renyi model, then that will give us a very nice penalty to try and encourage sparsity in our graph, which will lead to a sparse covariance matrix. Now, the Erdős-Renyi model is the most basic model you can have for a graph. What it says is each potential edge in a graph, for each potential edge, we draw a coin toss with probability beta. If the coin toss comes out to be heads, we draw an edge between the two variables. If the coin toss turns out to be tails, which happens with probability 1 minus beta, we won't draw an edge between the two. So the resulting probability distribution on the graph is beta to the power of number of edges times 1 minus beta to the power of the number of non-edges. And people have studied this model for graphs extensively in the network modeling literature, and the behavior of the Erdős-Renyi model crucially depends on the value of beta. If beta is less than 1 over v, the number of vertices on the graph, as v goes to infinity, the graph will be almost fully disconnected. It'll be very, very sparse. If beta is equal to 1 over v, as v goes to infinity, if it's behaving exactly like order 1 over v, you get kind of one component and then a load of singletons. When it's between 1 over v and log v over v, you tend to get a giant component and a few small disconnected nodes. And then once beta is bigger than log v over v, the graph becomes connected as v goes to infinity. So with this penalty, which I'm defining implicitly in terms of its exponential, the choice of beta will allow you to control the sparsity you get in the graph that you're using to model each mixture component very, very easily. In terms of if you choose a very small beta, you're going to get a very sparse graph. A moderate beta, you might get a lot of variables connected, but some variables disconnected. And then if beta is sufficiently large, you're going to get a full covariance matrix or close to a full covariance matrix. It won't necessarily be fully connected, but every variable will be correlated with some other variable. Similarly, in the network modeling world, people talk about power law distributions, and they tend to encourage kind of tree-type structures in graphs. So we propose that as another family of penalties you could potentially use if you had reason to think that your covariance matrix may have a covariance structure of this type. So we were kind of taking results from network analysis to come up with suitable penalties to get sparse covariance matrices in model-based clustering. So it's kind of combining the area of network analysis with model-based clustering and Gaussian mixture models to try and get sparse covariance matrices. And a final penalty we proposed is there may be some scientific reason why you would expect your covariance matrix to look something the non-zero elements of your covariance matrix to have a graph that looks very like a particular reference graph. So we proposed another penalty you could have is that you have some distance between the two, your estimated adjacency matrix and the reference adjacency matrix, and you penalize if they're very different from each other. You can use this approach to actually shrink the graphs for each cluster towards each other by assuming a common reference graph for all of the mixture components. So if we have just a common A star in our penalty term, it's going to force all of the graphs to look somewhat like each other. So just to start looking at some examples of this. So we now have, I'm just going to recap by going back a small bit. We have our model. We're going to be basically maximizing the likelihood of our model. We're going to choose a penalty, and then we're going to look at what clustering results we get. And I've talked about the algorithms and the types of penalties we can use to get that, and see what type of covariance matrix we get. So the first example I'm going to look at is data from breast cancer. So we have data from a breast cancer study where there were 5 different types of females that had 4 different types of breast cancer, and then normal, which are patients that don't have any breast cancer. So we've luminal A breast cancer, luminal B breast cancer, HER2 breast cancer, and basal breast cancer. So we have 5 different types, and for each woman, there was samples were taken, and they looked at genetic information on the samples, and basically there were 198 women, and we have 43 genes for each woman. So this is the raw data shown to us. So we're going to treat this as a clustering problem. So we know some of these women have different types of breast cancer, but I'm going to ignore that information. I'm going to do an unsupervised analysis. And what I want to see is what clusters am I going to find in my data if I cluster these 198 women on the basis of these 43 genes. Now, even though 43 genes is not very high dimensional, if you were to fit an entire full covariance matrix to each component of your Gaussian mixture model, it would have roughly 40 squared over 2 parameters just to model the covariance within each cluster, approximately of that order. So you'd have a lot of covariance parameters already, even in a moderate dimensional example like this. So, this is just an example that if you're trying to do Gaussian mixture models on data, you can't do it really with very high dimensional data. And we're going to see how well we do. We fitted a few different models to the data. We took the MCLUS models and we only looked at the diagonal ones because we weren't able to fit any of the MCLUS models that didn't have or that had off-diagonal elements that were non-zero. And that's because MCLUS goes from diagonal to having a full covariance matrix in kind of one jump. It doesn't have any kind of intermediate step. So we have, that got us an adjusted RAND index of 0.56. So the adjusted RAND index is a measure of agreement between the clustering results and the known breast cancer types that these women came from. 1 is the highest value the adjusted RAND index can get. And if you get an adjusted RAND index of 0, it means you're getting the same as random, a random assignment to clusters. And you can actually get adjusted RAND indices that are negative, but we're not going to see those happen here today. So 0 means you're only doing as well as if you randomly put the women in groups. 1 means you're getting a perfect classification. And then, it's quite a tough penalty to get close to 1. I will say that as well. It's very difficult to get an adjusted RAND index that's very high in most practical applications. The diagonal matrix model has 434 parameters in total. When we we did reasonably well with that model. When you do a full covariance matrix model, you can't actually fit that model to the data. When we did a sparse model using BIC, we got an adjusted RAND index of 0.6, and our model ended up with 1,063 parameters. With the extended BIC algorithm, it penalized, this is going to force us to get a much sparser graph than BIC because we set gamma equal to 1. We actually got slightly worse clustering results. Our adjusted RAND index is 0.58. But we massively reduced the number of parameters from over 1,000 to just under 500. And then the model that actually got the best adjusted RAND index was the sparse model that used the Erdős-Renyi penalty, and basically got an adjusted RAND index of 0.61, and it used 723 parameters. So using these penalties that we used from network analysis, where we said that e to the power of minus our penalty is the same as a network model that's commonly used, we actually managed to get better clustering results and an intermediate number of parameters between what BIC and EBIC was getting. And here's a table showing you the correspondence between the breast cancer types are the rows, and the clusters are on the y-axis, the vertical columns. And you can see that we're doing quite a good job separating the breast cancers into their correct types. A very nice output of this model is we have a graph for each showing us the relationship between the variables in each of the clusters. So the first cluster in the data set was 100% basal breast cancer women. And basically, we now have a graph showing us which variables in this data set are correlated with each other. So the model is telling us that, say, for example, I'm just going to focus in on the genes that I'm up in the top kind of left-hand corner. So we can see that SF4P1 is correlated with FOXA1, and FOXA1 is correlated with KRT5. And KRT5 is correlated with KRT14, for example. So we can see a structure in the correlation of the genes within cluster 1, and that cluster is mainly basal breast cancer patients. I'm going to look at the middle bottom graph next. And I kind of did look at this earlier, so I know what to look for. We can see SF4P1 is kind of near the top of the graph here. And it's very correlated with things like KRT14, again, KRT5. We have a very kind of highly connected component in the, and this cluster is mainly Lumina A and normal women, 89% luminal A women and 10% and 11% normal. And over on the right-hand side of the graph, FOXA1 is not actually correlated with any other variables. So we can see, I'm just focusing in on genes that I highlighted in the first cluster, there's quite a different correlation structure between those genes in the luminal A women. And if we look up at the top corner, which is a different luminal A, it's another cluster that's dominated by luminal A women. We can see that there's a disconnected small component at the bottom, SF4P1, KRT5, FOXA1, KRT13. They're all appearing together again in this luminal A group. So it's actually subdivided the luminal A women into two quite different groups here. And then for the two remaining clusters, we also have quite different covariance structures. And these covariance structures then are potentially of interest to the biologists so that they can see, you know, potentially there might be some idea about what pathways are going on in breast cancer, and it may give them some ideas about the differences between different types of sorry, I just noticed mute, just got it there. That's a nice output that you get from these sparse covariance matrices, that you can probe the different covariance structures in the different clusters. Just go back in here, sorry, my computer there. This is also just to show you how sparse the covariance matrices are. I just did a little heat map where I put a black dot between any two variables that are correlated within a cluster. So rather than showing it as a graph, and a white dot if they're not correlated, in the, like, so if the covariance is 0 between the variables. And you can see that only a very small fraction of the covariance matrices actually have non-zero elements. Most of the elements in the covariance matrices are quite are 0. So we're getting very sparse covariance matrices with this approach and with this penalty that we chose. A second data set is actually a lower dimensional data set, but it just kind of demonstrates the approach again. This is looking at a data set from patients that have hypothyroidism. So basically, endocrinologists collect data on patients that potentially have thyroid problems. So the patients fall into 3 groups. We have people who have hypothyroidism, which means that the thyroid is underactive. We have patients that have euthyroidism, which is that they have a normal thyroid. And then we have patients that are hyperthyroidism, which means their thyroid is overactive. For each patient, there were 5 different laboratory tests that were recorded. And the variables are called T4, T3, RT3U, TSH, and DTSH. And they're measuring different chemical measurements about the activity of the thyroid. If we apply our methodology again here, we found that we got a very good clustering of the data. We got 3 clusters that actually very closely resembled the 3 different diseases. So cluster 1 is very much dominated by people with an underactive thyroid. Cluster 2 is very much filled with people with a normal thyroid, and cluster 3 is people with an overactive thyroid. And again, we get very different graphs in each of the clusters, and we can see anywhere there's an edge, there's a correlation between the variables in that cluster, and if there's no edge, they're not correlated. We can see, for example, that T3 and T4 are correlated with each other in all 3 clusters, but then cluster 2 and 3 have an edge between T3 and RT3U, but the other cluster doesn't. So we can see that there's actually quite a difference in the covariance structures within each of the groups. So that was kind of a demonstration of a methodology that we've developed that gives very interpretable graphs and covariance matrices when you're doing model-based clustering, when you move into higher dimensions. It gives us a way of doing model-based clustering in high dimensions where we get a sparse covariance matrix, which isn't guaranteed by the other approaches. And it allows us scale model-based clustering to high dimensional data without making some unrealistic assumption like a diagonal covariance matrix, which would tell us that all of the variables within a cluster are uncorrelated with each other. We had to develop algorithms to implement this approach that allowed us to search over graph structures at speed. So I mentioned a genetic algorithm for doing this, but we also developed a greedy algorithm that's described in the paper as well, which allows us scale to even higher dimensions than the ones I showed for illustration purposes in this talk. If anyone here uses OR as a software package, I know a lot of people in kind of engineering and language processing are more MATLAB and Python users, but in OR, we've developed software which we called MixGGM, so mixed Gaussian graphical models, mixtures of Gaussian graphical models, and that's available to download freely. It's open source software for the OR programming environment. And here are some relevant papers, just kind of to talk through them very quickly. There's a paper by Charles Bouveron that looks at different model-based clustering approaches for high-dimensional data. This is the algorithm by Sanjay Chowdhury, Matthias Durton, and Thomas Richardson on estimating covariance matrices with prescribed zeros. The paper by Michael Fock, myself, and Lucas Gruca in statistics computing covers what I presented today. And then these are other relevant papers on graphical models. So thank you very much. And do people have questions?
Thank you very much, Brendan. It was very, very interesting. I see there is
I see a question on the
yeah,
I can see
there
the
is a
discussion.
question of Ran, and he asks, did you compare with neural network models?
We didn't, because well, we treated this, even though the examples I used have known groups, we actually analyzed the data as unsupervised, so as a clustering problem. So if we treated the problem as a as a supervised problem, we could directly compare it with neural networks. We haven't actually, even though actually the supervised version of this algorithm is much easier than the unsupervised, we haven't actually implemented, or we have actually written code, but we've never actually tested the supervised version of it. So no, we haven't compared it with neural network models because we were mainly focusing on the unsupervised kind of clustering approach. But if it was a supervised problem, it would be, neural networks would be a direct competitor. I could see that straight away, but we haven't done that because we focused on the unsupervised. And we focused also on generative approaches. So the models we're proposing here are generative models. So we're modeling the data generation process directly.
Thank you very much. I have also two questions to you.
Yeah.
First of all, if I understand correctly, your algorithm assumes that the numbers cluster is known in advance.
Not necessarily, because we use BIC to choose that as well, or we use the criterion to choose that as well. So we would have a BIC type penalty to choose the number of components. So what we would do is we would fit the model with 1 cluster, 2 clusters, 3 clusters up to some specified upper limit. And then we can use the criterion as well to choose the number of clusters. I didn't actually make that clear in the talk, but no, we can actually. But we do it by fitting different numbers of clusters and using the criterion to choose between them.
Okay, thank you. And another question, in the results of breast cancer, you show the results with beta parameter, with extended BIC, with extended BIC. It was for some beta that you defined. The best beta, or the best beta should be 0, because then it will be BIC. So how you choose the beta when you show the results?
I'm actually trying to remember. For the Erdős-Renyi, I actually would have to. I actually can't remember. I'd have to look at the paper to remind myself exactly. Well, in the. Let me just go back to where I've messed up my share screen here, I think. Yeah, let me go back. So for comparing eBIC versus BIC, so that had, let me go back to the penalties. Yes, so I see. So it was gamma, yes, so that's it, yeah. The results I showed compared BIC to when gamma is equal to 1. I went for the 2 extremes. So BIC penalized a lot less because when gamma is equal to 1, you're obviously getting a much bigger penalty on the number of edges. Yes.
So maybe gamma in between could give you a better result than BIC.
Yeah, I think it didn't in practice, but it could. In some applications, it could. Yes, it could.
Okay.
But when we did the Erdős-Renyi, I can't remember. I actually have forgotten how we chose beta, because that's another parameter where you need to, if you choose it, you're encouraging different types of graphs to occur. So you could try to choose the best beta, but it's just escaping me right at this moment what we actually did in that example.
Okay, Zev has a question.
Yeah.
Okay. Zev, please ask.
Sorry? Oh, yeah.
I tried to make unmute for Zev, and I don't know.
Yeah.
Can you help me to?
I can see it. There's a hand up. Zev is. He's still muted, I can see.
Yeah, one moment, we try.
Yeah. I think I can, oh yeah. Yeah, you can talk now, I think.
Oh, can you unmute him, Brendan?
Yes, I'm trying to find him. Yes, I think I can. I asked him to unmute.
They've disappeared.
He's a panelist now. He's appearing in the panelists
Ah,
window.
okay.
Yeah.
Can you unmute? Zev, you can ask the question. Zev, please ask the question. Okay, I think that Alberto also wanted to ask a question.
Yeah.
Can we unmute Alberto?
I'll try that now. Yeah, I'll try it. I've asked, yeah.
I have no question.
Oh, okay.
Okay. Okay.
Will I mute him
Okay.
again? I'll mute you again, Alberto.
Yeah.
Can you unmute Zev, please?
I've asked him to unmute. He must click something, I think, to allow it. Or if he wants to type it in the chat, I can read it.
Zev, can you type the question? Okay, we have a technical problem. We cannot unmute Zev somehow.
Yeah. I can actually, while we're trying to unmute him, I've actually opened up the paper and I can answer the question on how, for the Ur-Reni, what we did for the Ur-Reni model was we let the, looking at this, we let the penalty, I have it now, sorry. We let the penalty kind of proportional to, I'll go back to the examples. We went for kind of the upper end of this range, basically. So we allowed the penalty to be kind of log V over V. Was we kind of went for that to allow to kind of fall into that range. So we allowed it to be quite a sparse covariance matrix, but still has a lot of variables connected. So somebody was asking about that, the Ur-Reni as well.
Okay, thank you very much. We apologize for the problem, technical problem.
Not at all. Yeah,
we want to applaud you.
Thank you.
But we are all mute, but thank you very much.
Thank you very much. Thanks very much for the opportunity to talk to you. And sorry I didn't get to visit you in Israel this year.
Yeah.
So thank you very much.
Thanks again.
Great. Thank you.
Okay. Thank you very much to Brandon for his very, very interesting talk. And the next speaker will be Yoni Eder from Ben-Gurion University. He did his BSc at Ben-Gurion University Electrical and Computer Engineering. And now at the same department, he's doing his master's. Yoni's research interests include direct detection theory, statistical signal processing, and robust statistics. And his talk will be on measure transform to sample location testing. Okay, Yoni, please.
Thank you. Okay, so my name is Yoni Eder. I'm about to finish my master's degree in electrical and computer engineering from Ben-Gurion University. Today I will present my latest work under the supervision of Dr. Kobi Todros, entitled Measure Transform to Sample Location Testing. I will start with the background and motivation for the considered problem. Then I will briefly explain some basic principles on a probability measure transform. Via this transformation, I can elaborate on the method we developed, the measure transform two-sample Hotelling test, or in short, the MTHT. I will provide simulation examples from a fully generated data, as well as a demonstration of anomaly detection problem in a noisy video stream. And I will end up with a summary. So we're discussing the problem of detecting differences between multivariate population by testing for equality between the location parameters of their two unknown distributions. This test can be applied to many multivariate change detection applications. Wireless communications, DNA copy number alterations, neuroimaging analysis, and clinical trials are examples of such. As I mentioned at the outline, in the last part of the presentation, I will show how this problem can also be related to anomaly detection in a noisy video stream. So a natural test for this problem is the two-sample Hotelling test. This test, which only involves standard sample mean vectors and covariance matrices, has attractive implementation simplicity and tractable performance analysis that arise from the convenient normality assumption. However, a large deviation from these assumptions can lead to poor decision performance, for example, in the presence of heavy tail distributions that produce outliers. To overcome this limitation, several extensions have been proposed in the literature. First, a special rank test was proposed as a robust alternative. Additional tests that replace the standard sample mean vectors and covariance matrices with robust location and scatter estimates are the trim-Wins and Windsorized covariances, the MCD estimators, and the M estimators of location and scatter. This test may be successful in mitigating the model mismatch effect. However, this may result in degraded performance under Gaussian data, as well as increased implementation and computational complexity. In this work, we developed a new robust extension of the two-sample Hotelling test, which operates by applying a transform to the probability measures of some reshaped versions of the two compared datasets. This method compares simple and robust estimates of the location parameters theta1 and theta2. These are conveniently obtained from the empirical measure transformed mean vectors of the reshaped dataset. The measure transformations are adapted to the underlying distribution through a convex optimization procedure. This procedure approximates the maximizer. I have a problem with the mouse. Can I do a laser point?
No, they will not see the laser point.
Okay, so I'll cancel it. And they can see my mouse. Okay, so here I will review some basic principles of the probability measure transform. Given a non-negative function u from the vector space Rp to R+, which satisfies this regularity condition, a transform Tu is defined as this integral of phi of x, where phi of x is defined as the function u of x divided by its expectation under Px. The function u is called the empty function. By modifying this transformation, a wide range of transformations can be obtained. The mean vector and the covariance matrix under the transformed measures can be expressed as weighted mean and weighted covariance under Px, where phi of x is the weight function. They are called the empty mean vector and empty covariance matrix. These statistics universally take finite values whenever these conditions hold. Given a sequence of n samples from Px, the empirical empty mean vectors and empty covariance matrix are defined as follows. If the samples are mutually independent and this expectation is finite, then the empirical empty mean vectors of x tilde and the empirical empty covariance matrix are strongly consistent estimators. So we're discussing the problem of testing for equality between the location parameters of two unknown symmetric probability distributions, Px and Py. Data samples available from each distribution satisfy this observation model, where θ1 and θ2 are the location parameters of Px and Py. Wn and Zm are statistically independent IID random processes with unknown symmetric distributions about the origin. Note that we do not assume that the noises follow some known distribution. Hence, we consider the composite binary hypothesis testing problem, where the null hypothesis is defined when the location parameters are equal, versus H1 when they are not. We define a reshaped model by concatenating each even sample before its previous, which produce the random vectors xn tilde, ym tilde, wn tilde, zm tilde, along with the matrix A. Note that there is no statistical dependency between the concatenated vectors. Following this, we apply the transforms tu1 on px tilde and tu2 on py tilde. One can verify that for any pair of functions, empty functions, that satisfy these conditions, we obtain that the empty mean vectors of x tilde and y tilde satisfy these simple linear relations. As a result, we can easily define those estimators of the location parameters θ1 and θ2 based on the empirical empty mean vectors of x tilde and y tilde. So based on the last estimators, the empty HG is defined as follows, where B hat is an empirical estimate of the asymptotic covariance matrix of this scale difference under the null hypothesis. And the matrix B hat is defined as follows. The empty HG is invariant to translation of the considered datasets. Rotation invariance is guaranteed whenever these conditions hold. Robustness to outliers is induced by outlier resilience of these 6 Fisher-consistent statistics that comprise the test. These statistics are B-robust whenever the empty functions are bounded, as shown here. To analyze the asymptotic performance of the empty HG, we assume that the following regularity conditions hold. We showed that the asymptotic distribution under the null hypothesis of the empty HG is chi-square distribution with p degrees of freedom. This property allows us to obtain an asymptotic threshold to maintain a constant probability of false alarm, that is, a CFAR detector. Consider a sequence of local alternatives where θ1 is in the vicinity of θ2. Then, the asymptotic distribution of the empty HG under H1 is non-central chi-squared with p degrees of freedom, and non-centrality parameter defined as this random variable whose entries are defined here. So, the asymptotic distributions of the test under both hypotheses allows us to obtain the asymptotic local power at a fixed asymptotic false alarm rate alpha. So how did we choose those empty functions u1 and u2? We showed that the minimization of this trace of the matrix B with respect to u1 and u2 amounts to maximization of a lower bound on the asymptotic local power. Moreover, when the empty functions belong to some linear space of functions, the problem of minimizing this trace with respect to the positive vectors a1 and a2 is mathematically equivalent to the following quadratic programming problems, where the statistics of the optimization problem are defined here. Obviously, their true values are unknown. Therefore, we replace them by their empirical estimates. Okay, notice that both the estimation and optimization procedures are performed on the data itself. We do not use a training set to achieve them. Okay, so now I will show a simulation example where the observations from Px are derived from a symmetric GMM distributions with constructed by 4 mixture components with equal probabilities, while the measurements from PY follow a t-distributed noise. In addition, consider these following settings. This figure depicts average shapes of the optimized empty functions U1 and U2. One sees, as expected, that the empty functions U1 adapted to the light-tailed GMM data is significantly wider than the one adapted to the t-distributed data. Here you can see a computational complexity assessment of the empty HD compared to the classical Hotelling test and other robust extensions. Unlike other methods, our method is linear in the sample sizes n and m. In addition, its iterative part, as can be shown here, it's independent of both the sample sizes and the dimension of the observations p. And this assessment was verified by an examination of average simulation running times of the considered test versus the dimension of the observation p. The sample sizes increase accordingly. You can see that our method has the lowest runtime values compared to all other robust extensions examined. This figure depicts an RC analysis of the considered tests. An approximation of the asymptotic local power was added in black, along with the GLRT in gray for a benchmark, which the GLRT utilized prior knowledge of the distributions of the datasets. You can see that our proposed method in blue outperforms all other robust extensions, coincides with the asymptotic MTHD, and reduces the gap towards the omniscient GLRT. In addition, you can see in gold that the MCD HD, which is based on the popular MCD estimators, has poor detection performance. As can also be seen in this analysis of detection probability versus SNR. This degrading performance is due to the MCD estimators, which in the case of high SNR are based on measurements from a particular cluster rather than all measurements given. And also here you can observe the superiority of our method compared to others. So after all this theoretical analysis, we finally reached the example which is based on a real video stream. Our goal is to detect an intrusion attempt in an office environment where the data is obtained from the following video sequence. Okay, note that this video was accelerated due to our time constraints. It's supposed to be 10 times faster, slower, sorry. So, as you've seen, the break-in attempt moment is represented by a change in the angle of the door handle. And this problem is not very difficult when the video is of good quality. It is more challenging to examine the case where the video is blurry and noisy, especially in the time window of the break-in attempt. So in the experiments we created, first we slightly blurred all the video frames. Then we set up a reference frame, which is a relatively high quality frame at routine, meaning that it was noised by a light-tailed Gaussian distribution at most. Next, we added heavy-tailed noise distributions to all other video frames in order to raise the level of difficulty in detecting the intrusion attempt. One can see, yes, okay, sorry. To simplify the problem, we will focus on this region of interest. So, you can notice that whenever the image is still, it can be divided into blocks which are roughly homogeneous in color. Let's look at this block for example. This block contains the 3 RGB layers, each with n pixels. This structure can easily be transformed into a set of n 3D vectors which are dimensionally correlated. Now, due to the homogeneity in the pixel values, adding an IID noise to this array will result in a good approximation of our considered observation model. Hence, we can compare the reference frame with all other video frames by converting each block to the observation model and using our proposed method on the reshaped parallel blocks. Now, after testing each parallel subframes, we take the maximum value of all the results obtained. In this example, we added a Gaussian noise to the reference frame with the following settings, and a t-distributed noise of the following type to all other video frames. This figure depicts the empty HD and the HD values relative to the reference frame over time. You can see that our method in black responds to the intrusion attempt that occurred between these two vertical yellow lines, as opposed to the standard hoteling test. In addition, the empirical threshold coincides with the analytical one, enabling a fine detection rate. By using these videos, we were able to produce a reliable ROC curve. And also here, you can see that our method outperforms all robust extension examined. So, to conclude, in this work, we developed a new robust extension of the two-sample hoteling test by applying a probability measure transform. We showed that proper selection of the involved empty functions can lead to a simple and robust test that outperforms the hoteling test and other robust extensions. This test can be applied to many change detection applications that contain outliers. As a further study, an extension can be made to more than 2 samples given, as well as studying the case of non-symmetrical noise distributions. Thank you very much. And this is my email in case you have any questions or want to know more about my research.
Okay. Thank you very much, Yoni. I don't see questions, so I have one.
Okay.
You show here that you compare Gaussian noise and heavy-tailed noise. Can it work with other distributions like Laplace or gamma distribution, and should it be only unimodal distribution, or it can work with other type of distribution like GMM that was presented in the previous talk?
Yes, definitely. It is important thing that we do not assume that the noise distributions follow some known probability function. Therefore, we can apply our method to many type of distributions. And I don't see any reason why it shouldn't work on GMM as I showed in the simulation study. So that's it.
Okay, thank you very much, Yoni.
Thank you.
Again, for a very interesting talk. And this is our last talk.
Okay, thank you very much.
Okay, thank you very much for the speakers, Brendan and Yoni. And thank you very much to all the audience. It was our first webinar for unsupervised machine learning, and I hope that it will be the last one, and next time we can do it in live, not via web, and we will see you all at Afeka. And here I conclude the seminar. Wish you all good health and see you next time. Goodbye.